We're going to start these lectures with a cell biologist's view of an animal life cycle. As you all know, it starts with an egg and a sperm. These are usually the biggest and the smallest cells, uninucleate cells at least, in the entire life cycle of an animal. And they are haploid gametes. That is, they have one copy each of a parental genome. They get together and they fuse to form a zygote, which undergoes, the zygote is diploid, the diploid product of gamete fusion, and that zygote undergoes cell division, mitosis, the end product of which ultimately is a bunch of cells in an embryo or a body of some sort. Those cells look something like this. They're diploid. This is an epithelial cell. Which is typical of many of our tissues, at least the parts that form sheets. It's polarized, it bears a cilium, much like the sperm. And ultimately, the fate of this cell is to produce in turn eggs and sperm, that is, more haploid gametes. For diploid cells to produce haploid gametes, they go through meiosis, which I'm drawing here as a very unequal cell division, which it is in the case of the meiotic divisions that make eggs, although not so for sperm. At any rate, this boundary marks approximately the division between what we call the germline, germ cells, gametes, and the soma, somatic cells, the body, soma is body. Of course, there might be a variety of other things in a body besides simple epithelial cells like this. One straightforward thing that epithelial cells might do is to become motile, to crawl. Transformation to what we call a mesenchymal cell is usually a reversible one. Animal development is full of so-called epithelial mesenchymal transitions, EMT, but it is also full of less reversible events. Cells might differentiate to adopt various structural or functional states, such as, for example, a relatively straightforward modification of the simple epithelial cell in this cell. That cilium has been greatly elaborated into this fir tree-like apparatus. Some of you may recognize this as an iconic version of the cone cells in the retina. This happens to be a photoreceptor cell type. So the germ cells and the somatic cells are united by repeated cycles of cell growth and division, punctuated by sex, which means this special kind of division followed by cell fusion. Somatic cells might become motile, or they might split off from the cell cycle to adopt functionally and structurally differentiated states, multicellular life forms like kelps or mushrooms or animals differ from unicellular eukaryotes chiefly in the fact that what we're calling somatic cells tend to hang together to form large bodies that conduct most of the actual business of living, often in the case of animals. Some fraction of somatic cells, pretty much 100% for most animals, give up their potential to become germ cells by differentiating to make functional tissues and organs. Then there's a distinction between groups in which the body, where most of the life cycle happens, is a product of meiosis or a product of gamete fusion. Again, for us, it's the products of gamete fusion that are walking around and talking to each other. Although for some seaweeds or other multicellular life forms, it's actually the haploid cells produced by meiosis, which form a body that then goes on eventually to make gametes. And the zygote might be very short-lived. Finally, animals are distinguished from the other multicellular life forms on this planet at the moment simply by the immense variety of differentiated cell types that they might adopt in the course of the life cycle. Now, I'm going to draw an epithelial cell in a little more detail, and I'm going to draw a stylized cell that really exists, and more or less as I'm going to draw it in many organisms, and in modified form constitutes kind of a ground plan for many of our differentiated cell types. So this collar cell, first of all, is epithelial, and as such, it's going to have neighbors, but I'm not going to draw them in detail. It's going to have neighbors to which it's bound by junctions. That is, of course, how our cells hang together to make tissues. They have to have some way of recognizing each other and sticking to each other stably. Okay, now it has a bit more apparatus. Of course, it has that cilium. Many of our cells bear beating cilia. Those that don't, many of them bear what's called a primary cilium that doesn't beat. And it's surrounded by, the reason it's called a collar cell is it's surrounded by a collar of microvilli, fancy word meaning little fingers. The cilium, as we'll learn about, is based on microtubules, one of the great cytoskeletal polymers that makes eukaryotic cells work. The microvilli are based on actin filaments. The cilium, incidentally, is the same for eukaryotic cells as the flagellum. These two words refer to the same structure, distinguished in common usage only by whether the cilium grows long enough to have a sinusoidal beat, a sinusoidal waveform to its beat, in which case we call it a flagellum, such as the cilium borne by a sperm. Okay, anyway, so these two surface projections represent typically distinct cytoskeletal elements, one based on microtubules, the other based on actin filaments. And now I'll proceed to fill in a few more details before proceeding. All right, again, we have junctions by which cells are coupled to their neighbors. This green object here is the so-called basal body, from which grows the skeleton of the cilium. The basal body is also known as the centriole, and we'll talk about its transformations. It turns out it is one of the key contributions of the sperm at fertilization. Of course, there's a nucleus in here that contains the genome walled off from the cytoplasm. These yellow things are microtubules, whereas the red things up in the microvilli are actin filaments. Microtubules typically extend through the cell, and by organizing traffic within the cell, give it polarity. This cell has apical end facing the outside world, typically, in some sense, versus the basal end. And this layer of purple matting down here is likely secreted by these cells, shaped by them. It is their extracellular matrix, or ECM. And in this case, it often goes by the name basal lamina. Get it? Because it's a lamina, a layer, and it's on the basal side. Okay? So these basic features of an epithelial cell type can be found in a variety of our other differentiated cell types. But first, I want to go to the other side of the board and make a comparison between this cell type and our closest unicellular relatives, the choanoflagellates. Our relationship to choanoflagellates was realized by zoologists in the 19th century who noted the structural and functional resemblance between these unicells and the cells within sponges that pump water through the sponge body, the so-called choanocytes. Choanoflagellates are not multicellular, although many live in colonies. The cells are functionally autonomous, and they're characterized by a cell body that bears a long beating flagellum surrounded by a collar of membrane extensions. We would now call these microvilli. And these microvilli are in fact supported by actin filaments, just like the ones in the stereotyped collar cell that I drew. Of course, there's a nucleus in here and a basal body anchoring that flagellum. The flagellum here beats so as to make a water current going that way, which of course means water is being transported past the cell body, and most importantly, past this veil of membrane supported by the collar of microvilli around the flagellum. The water current might be carrying objects like, say, bacterial cells, and the function of this collar is to intercept those bacterial cells and engulf them as food. So choanoflagellates make a living by driving a water current and filtering it by intercepting small particles, bacteria, on a collar of membrane supported by actin-rich microvilli. And that structural relationship to many of the cells in animal bodies is elaborated in a variety of differentiated cell types. The first one that I'm going to mention is an olfactory receptor. It takes a few liberties with the structure of an olfactory receptor, but you'll find in textbook diagrams of an olfactory receptor a collar of microvilli on the apical end of the cell, maybe some projections connecting it to nerve cells or something like that. Most olfactory receptor cells bear several long cilia which they extend into the surrounding fluid. The cilia, in this case, behave as antennas, not so much as motors. They behave as antennas, sensing odorants, suspended chemicals in the medium in which that passes over the cell surface. So that's a fairly straightforward elaboration of the collar cell type. Another, I would say, relatively straightforward elaboration is this cone cell photoreceptor over here, in which case the cilium has simply become swollen into this photoreceptive antenna. Another simple, seemingly simple transformation of this basic cell type is to have multiple cilia, an epithelial cell with many cilia growing from its surface. This is typical of almost all of our fluid ducts, the airway, fallopian tubes, the brain ventricles, etc. Now, it's not as simple as it seems, because in animals, there's a key step that's taken when cells decide, as these two have, to make more than one cilium. Each one of those cilia needs a basal body, and for reasons we'll discuss in detail but aren't actually completely clear, once a cell makes multiple basal bodies, it can never divide again. Once an animal cell makes multiple basal bodies, it can never divide again. Much of the rest of the eukaryotic world doesn't seem to have any trouble with this, but it's a major constraint for animals. So here we have, again, an olfactory receptor. A multiciliated epithelial cell. Let's consider some really highly unusual cell type, like a neuron. Well, actually, it's not that much different. The neuron has a cell body that, as many of you know, might include numerous projections, dendrites, that serve to connect it to other neurons. And then typically one long axon ending in a nerve terminal by which it connects to, by which it synapses to other nerves or perhaps to a muscle in the case of a motor neuron. The axon is based on a thick bundle of relatively stable microtubules. It's not really the case that one single microtubule runs from your spinal cord all the way down to your big toe, but you can pretend. At any rate, these microtubules both support the axon structurally and conduct traffic along it, as we'll see. Dendrites, by the way, are also supported by microtubules. So this cell, like this one, is polarized in at least one axis. As it happens, many neurons still bear a beating cilium. If that doesn't make you tickle, it should. And one can think of the axon roughly as a greatly elaborated apical specialization. I mentioned before a more radical departure is the conversion of epithelial cells to mesenchymal cells. Draw it over here in profile this time. That means, of course, losing the junctions that characterize animal tissues, typically, and instead making junctions to some sort of extracellular matrix. Examples of these include the fibroblasts that remodel tissues and craft the extracellular matrix between the rest of our cells and so on. Keratocytes that heal wounds. We'll read about keratocyte motility later in the course. This extension of the cell surface is actually the crawling entity that does all the work, the lamellipod. Again, we'll talk about how that works later in the course. Suffice it to say that in most mesenchymal cells, this lamellipod is propelled by the continual assembly and disassembly of actin filaments. So this is a neuron, of course. This is a mesenchymal cell again. A generic representation of many kinds. We actually still contain a cell type that functions not dissimilarly to choanoflagellate. In our kidneys, we have podocytes, which bear, again, a beating cilium, facing, in that case, the fluid that's being transported through the kidney for filtration, the filtrate in this case, beating into the primary urine. The cell surface in podocytes, this is a elaboration in a sense of the collar of microvilli. Podocytes are so named for these feet that they extend across a very dense extracellular matrix, so dense that it behaves as an ultrafilter. And we'll draw in that woven membrane, that woven extracellular matrix here. They're dense basal lamina supported on this lattice of microvilli. And while I'm finishing this drawing, try and think of the strangest cell that departs the most from this structure in your own body. I know you can't talk back because these lectures are recorded, but what is the very weirdest cell in your body? Okay, I obviously have some in mind myself. They are at the extreme ends of the scale, but they are not a sperm and an egg. Here's one of them. I'm sure you all recognize that as a red blood cell. A mammalian erythrocyte loses its nucleus, certainly loses its cilium, loses junctions, loses almost everything that there is to being an animal cell, except for a membrane and a few other associated cytoskeletal structures, and becomes just a bag of hemoglobin circulating in the blood. I would nominate as even stranger the skeletal muscle cell. Now, I can't even draw a skeletal muscle cell to scale here. skeletal muscle cells are enormous. Of course, they are what gives us rapid movement as animals. Skeletal muscle cells are dominated by dense arrays of contractile proteins, and I'm going to draw out a simple cartoon of one, starting with the repeating boundaries between subunits of the myofibril, that is, the contractile apparatus which muscle cells contain. These happen to be called Z-lines, as we discussed later, and they define the boundaries of sarcomeres. Don't worry about the vocabulary for now. We'll come back to it. We'll come back to muscle later on in the course. From the Z-lines grow ordered, dense arrays of microfilaments, actin filaments. Now, actually, the growing ends of these actin filaments are buried in the Z-line, but again, we'll come back to that later. Pardon me while I fill these all in, just for the sake of completion. Okay, these actin filaments are half of the contractile protein assembly. They're approximately half of what you're paying for when you buy a steak. The other half is the protein that does the actual mechanical work, myosin, the so-called thick filaments, consisting of many motors ganged together, are interdigitated so that their heads are between these actin filaments and can engage with them. Again, I'm going to fill this in for the sake of completeness. Okay, thank goodness for the speed up function, eh? Roughly, the way skeletal muscle works is that at rest, these motors, the blue things, the thick filaments, are prevented from engaging with the actin filaments. But in response to a stimulus from a nerve at the cell surface that makes a synapse there, the actin filaments suddenly become available for these motors to engage with. And within milliseconds they do, and they crawl along them, and some crawl this way, and some crawl this way. making this contractile unit shorten, perhaps by 20% or less for many muscles. So, of course, if it shortens all the way along, you get a muscle contraction out. Building this thing, just like it took me a long time to draw one, imagine takes a great deal of constructive effort on the part of the cell. And it's also quite large. What I haven't drawn in this cell is all of the other myofibrils populating this myotube. Typical skeletal muscle cell contains many of these fibrils stacked up in parallel to each other and aligned. And of course, physiologically, this cell must be able to coordinate the contraction so that instead of, some of these going off and then some of them going off over here and so on, they all go off together and work in tandem. That takes very many cellular specializations, which we'll revisit later on, but it also takes a great big cell that couldn't possibly be the work of one nucleus. And skeletal muscle cells are typically populated by very many nuclei. How did this get to be the case? And they might be sandwiched in here between adjacent myofibrils or tucked right under the plasma membrane or something like that. There might be hundreds in a large myotube. You might imagine that perhaps those nuclei proliferated within there, as some eukaryotic cells do. They might multiply nuclei within the cytoplasm. But in fact, skeletal muscle is typically created by the fusion of precursor cells, by the fusion of individual myoblasts to make this one giant cell. So I would nominate this as the very greatest departure in a typical animal from this epithelial model. Now, how is this all different from prokaryotes? If we take a look at one of these hapless little bacterial cells, it's a perfectly good cell. We look at it up close. It has a cell membrane that's physiologically quite active and interesting. There's a genome wrapped up in there somewhere. Often it's a circular genome in big loops. And of course, the reason for the different name for prokaryotes is that they lack a nucleus. But that's not all there is to it. Prokaryotic cells, of course, many of them can swim. They have a remarkable little rotary motor, many of them embedded in their membrane, which spins a protein filament that they can swim with. This bacterial flagellum is totally unrelated to the eukaryotic cilium or flagellum. It functions in a completely different manner than the familiar one that we use. The biggest difference between eukaryotes and prokaryotes is that most prokaryotic cells lack the kind of deformable membrane supported by a dynamic cytoskeleton that makes eukaryotic cell shapes so diverse and versatile. They also lack internal membrane compartments, which allow us to wall off various kinds of activities within the cytoplasm. The nucleus is only one of the many kinds of compartments that we'll have occasion to mention and visit. Now, this course is going to cover some of the essential traits of eukaryotic cells, how they change shape, how they divide, how they move. We're going to visit many unicellular organisms along the way, and also different kinds of multicellular life forms, but we're always going to return as a sort of central framework to animal development, partly because I know it best, partly because biologists really know it best. And those are the things that we are most likely to find detailed examples of in textbooks, and we will rely heavily in our readings on cell biology in developing animal embryos and larvae. In the next lecture module, we're going to start with a detailed look at fertilization, which is our biggest subject for this week.